inflammation and scarring, or fibrosis, are important responses to injury, but when excessive, they result in disease and death. I integrate structural biology approaches with cell and tissue studies to understand how these occur and how they are linked right down to the molecular level. I look at how genetic mutations and environmental stimuli affect protein conformation and interactions to cause disease, and how new drugs may target and prevent this. A protein called galectin-3 forms molecular lattices at the cell's surface in response to acute injury or pro-fibrotic signaling by the cytokine TGF-beta. We have shown how this clusters transmembrane proteins, including mediators of inflammation and mediators of fibrosis. We call this assembly the GAL3 fibrosome. Recently, we have interrogated its involvement in the severe lung inflammation that causes most death in COVID-19. But for the last few years, we've been studying how it enables and accelerates the fatal lung disease idiopathic pulmonary fibrosis, or IPF for short. In IPF, scar tissue gradually replaces lung tissue. Median survival with this is 3 years from diagnosis, and it accounts for 1% of all UK deaths. Disease progression is punctuated by sudden inflammatory flares known as acute exacerbations that accelerate decline and have high mortality. Current treatments help by slowing the rate of decline, but they do not cure, reverse, or even stabilize the disease, and there are no treatments proven for acute exacerbations. Targeting key features of the GAL3 fibrosome prevents the positive feedback between inflammation and fibrosis that is a motor of disease, whilst preserving important pathways like TGF-beta signaling in other contexts. So, we want to target GAL3 fibrosome constituents and assembly to treat fibrotic diseases such as IPF. Galectin-3 is a biomarker for multiple fibrotic diseases, and targeting galectin-3 rescues a wide range of experimental models of fibrosis in vivo. We have also successfully targeted other GAL3 fibrosome constituents using small molecules or gene silencing in models of acute exacerbations, blocking inflammation and the knock-on pro-fibrotic effects. I work both as a consultant in a major respiratory and translational research centre and as an investigator using the biophysical and structural capabilities here at LISCB. This means that I can directly cross-check findings from my molecular studies using biosamples from patients, including cultured lung tissue, and vice versa. As a doctor, I want to see the new therapeutic strategies that we've defined and proven concept in develop to their fullest potential for patient benefit. We've already attracted support from a company whose IND blocking galectin-3 is in phase 2b trials in IPF. But detailed characterization of the GAL3 fibrosome as a whole identifies a wider range of targets across multiple disease contexts with huge unmet clinical need. We would be delighted to collaborate with industrial partners to make inroads into this.